Okay, so now let's continue the chapter 4, the protein structure. Right, so in this chapter, we'll discuss the amino acid and also how the amino acid form the polymer to become the protein, and also hierarchical organization of protein structure and also protein folding. Right, so basically, the protein are the polymers of amino acid, and the amino acids are covalently linked together through the amine bond, which is the peptide bond that I will show you later in the slide. And the peptide, the short polypeptide, usually less than 40 amino acids in length, also called oligopeptide. Okay. And the hydrophobic effect, the hydrophobic amino acid tend to cluster in the interior of a protein. That's called hydrophobic effect. So all those hydrophobic protein amino acid, they will cluster in the interior protein. So like, for example, here we have this serine hydrophilic and aspartate the hydrophilic, and the cysteine hydrophilic amino acid, and leucine hydrophobic, and then if a long chain of amino acid, and then these hydrophobic amino acids tend to cluster inside the protein, okay, so that they can minimize the interaction with water. And the basic amino structure, so it's like this here. So we have the alpha carbon, right? Alpha carbon on the left side, amino group on the right side, carboxyl group. Okay, that's this is the basic structure, and then here we will have the side chain. So all the amino acids have the similar structure of this basic structure, the amino group, alpha carbons, carboxyl group, and hydrogen, and only the side chain is different. Okay, that gives you a different amino acid. And the arrangement of amino acid in a polypeptide chain that can determine the structure and function of protein. Okay? And the amino acid are the chiral. So basically, this is the basic structure, all right? However, they have a mirror image structure, can be the left hand, right hand, the arrow amino acid, the amino acid. And this basically is the location of the amino group and carboxyl group. So in the left hand alanine, for example, right? So you have this alpha carbon in the center, okay? And then the side chain project to the top, okay? This alpha carbon in the center, and then the side chain point to the top, and then the hydrogen to the back, and then on the left side amino group, on the right side is carboxyl group. Then this is the arrow form amino acid. And in the nature, most of the amino acids are arrow form, okay? And then this just to tell you they have different configuration. The S configuration, R configuration. And on the first column here, that's the commonly seen 20 amino acid, glycine, alanine, valine, leucine, isoleucine. The three-letter abbreviation is the glycine, GLY, alanine, ALA, valine, VALO, and then single-letter abbreviation, GLY is G, glycine, alanine, A, valine, V, leucine, L, isoleucine, I, proline, P, methionine, M, phenylalanine, Phe, and F, and then tryptophan, so it's W, and then tyrosine, Y, and then serine is S, and threonine is T, and cysteine C, aspartate acids D, and glutamine S is E, asparagine, and then glutamine is Q, histidine H, lysine is K, and then arginine R. Okay, so these are the single letter abbreviation for the amino acid. And then the amino acid can be classified in these four different groups. But normally we don't classify this way, but in this book, they classify the charged. So that's a positive and negative. This is 3 lysine, arginine, histidine, positive charge. Aspartic, glutamine is negative charge. Hydrophilic, right? Hydrophobic, all right? Aromatic, these phenylalanine, tyrosine, tryptophan, they have the aromatic ring. And these 3 are important to for the protein concentration determination, because the aromatic ring can contribute to the spectrophotometer, the fluorescence, so that you can determine the protein concentration. So the charged amino acid, aspartate, glutamate, right? They are charged. C is the negative charge, negative charge. So this is the amino group, carboxyl group, amino group, carboxyl group, and then alpha carbon, and negative charge, right? So these 2 are negative charge. And then these 3, lysine, arginine, histidine, positive charge. You see the positive charge, okay? And then possible the positive charge here, okay? And then hydrophilic. So these are the hydrophilic: serine, threonine, cysteine, hydrophilic amino acid, and cysteine. You see they have the sulfur here, and this is the cysteine is the only amino acid can contribute to the disulfur bond in the tertiary protein structure. Okay. And then hydrophilic. So you can see these hydrophilic here, they potentially can form these hydrogen bond with water. Okay, so there are five hydrophilic. It's a polar amino acids: serine, threonine, cysteine, asparagine, and the glutamine. They can form a hydrogen bond. Okay, but not usually charged at the physiological pH. So that's why we call these 5 amino acids are hydrophilic amino acid. Okay, so they are polar amino acid. Then so these are just to show the hydroxy group on the serine here, right? Can be phosphorylated by the enzyme, the kinase phosphatase to phosphorylate. See, hydroxy group phosphorylated. Okay. And the cysteine, okay, the 216 can be oxidated, right? Oxidated, remove this hydrogen, so they form this disulfur bond. They can stabilize the tertiary protein structure. And then 6 amino acids are hydrophobic: alanine, proline, valine, leucine, isoleucine, and the methionine. They have the hydrophobic property. Okay. And the leucine is the most abundant amino acid in proteins and play a major role in promoting the hydrophobic interaction in the globular protein. Okay, that's the leucine. And the 3 here, the phenylalanine, tyrosine, tryptophan. Okay, so these are the amino acid that has this ring. Okay, they have this ring. but in another classification, although they contribute to because the absorbance, they contribute to the protein concentration determination, and but mostly in some other textbook, they can classify phenylalanine as a hydrophobic, and then tyrosine tryptophan, both are the polar hydrophilic amino acid. Okay. So summary, we have all the 20 amino acid alpha amino acid. For 19 out of 20, the alpha amino group is primary. For proline is secondary. And the alpha carbon of each is a sterol center except the glycine, isoleucine, and threonine contain a second sterol center. Okay. So the ionization of amino acid. So amino acid, basically, they exist in neutral solution as a neutral ion with no net charge. Okay, no net charge, because the carboxyl group will be CO negative, and then amino group NH3 positive. So it's no net charge in the neutral ion. And at that point, the pH point at which there is no net charge on the neutral ion, we call that isoelectric point, okay, PI. So you can see these amino acid, right? The basic structure is amino group, carboxyl group, positive, negative, so it's a neutral, all right? If you go in this direction, going to the left, all right? Go in the left, you obtain the hydrogen for the carboxyl group to become positive. To the right, okay, you remove the one hydrogen ion, which is from this amino group, so from NH3 plus to the NH2, and now it becomes negative charge. So here's positive, here's negative charge, in the middle is net zero charge, zero net charge. And this point, the pH at this point is called isoelectric point. Okay, it's a PI. And here's the example of the histidine. So zero net charge. So you have the amino group, carboxyl group. Okay, if you go into the right, all right, going to the right, you remove one hydrogen ion from the amino group. So it'll be NH2CO negative. So -1 charge. And then if you go into the left, right, going to the left, going to the left, the first step is this nitrogen to accept the hydrogen. Okay, so that's the pKa 1, and then you have 2 here. That would be the real amino group and the carboxyl group to accept the hydrogen here. So here +1 and +2. So isoelectric point, which is the PI, that would be the pH at which the amino acid is neutral. Okay, so the PI here, for example, PI here is a neutral, be like this here, right? The neutral, we have positive amino group, negative carboxyl group. And going down, which is going this direction, pK1, right? pK1, so that the carboxyl group receive the hydrogen ion and +1. If you go up, all right, from PI to PK2, see the PI to PK2, okay, difference, you see the difference. So you lose the hydrogen ion from this carboxyl group. This is not from this the main amino structure. Okay, that's from the side chain. The side chain release one hydrogen. And then pK3 would be the hydrogen ion is released from the amino group. Okay, so that's a PI. And ionization amino acid, so we see the carboxyl group, amino group, okay? Remember here, remember to the left, carboxyl group, to the right, amino group. So for one amino group is higher pK, and then to the carboxyl group is lower PA, right? So lower one here, see the column, lower pH, that's the carboxyl group, and higher pH, that's the amino group. Okay. And then most amino acids only have 2 pKa value. So the equation is easily used to calculate PI because you just add 2.34 plus 9.6 divided by 2, that'll be the PI for the glycine. For alanine PI, this 234 plus 969 divided by 2. Okay, if they are two groups are dissociated at the isoelectric point, isoelectric pH, the pKa1 is the higher pKa of the two. And then pKa2 for the group that has not dissociated at the isoelectric point, isoelectric pH. So that means, see the cysteine, for example. You have this 171A33 10.7, okay? And then for the to calculate the PI, you would take these highest, that's for association of the amino group 10.78 and plus the carboxyl group 171 divided by 2, and then now calculate this one in the middle, okay? So you have to take the one from the carboxyl group, one from amino group, and divide it by 2. That's how you determine the isoelectric pH, okay? Formation of peptide bond. So basically formation of peptide bond, it's a condensation. So it's a dehydration, okay? Dehydration. So this is the first amino acid, second amino acid. Then the carboxyl group of the first amino acid will react with the amino group of the second amino acid. And then you release one molecule of water, and then to form this CONH here, okay? That's how they form the peptide bond. And the genetic code, that means that explain the relationship between RNA and protein. Okay, so the genetic code. So you read from the first letter U, second letter U, and then the third letter U, the UUU, that's phenylalanine. Okay, that's genetic code. So basically, every three nucleotide, that can help to determine the amino acid. Okay, so the RNA genetic code identify amino acids specified by 61 out of the 64 possible triplet codons. Okay, redundancy exists in the RNA genetic code because multiple triplet codon can specify the same amino acid. So like, for example, here protein, the CCU, CCC, CCA, CCG, they can also define the protein. Okay, that's called redundancy, right? And then we also have a stop codons, UAA, UAG, and the UGA, okay? And also the wobble effect, wobble base, that mean, like, for example, CCU, CCC, CCA, CCG, these four on the third position of the codon, all right? That's the wobble effect, because the tRNA, when they bring in the amino acid, they'll be able to identify these four nucleotides, okay, with the same tRNA, and so that gives you the protein. Okay, that's a kind of a wobble effect. And then the DNA bioinformatics can predict the amino acid sequence. So basically, from this DNA and RNA, from the triplet genetic code, then we can predict the amino acid sequence. Okay. And then for the peptides, to determine the pI, the pH, and for the peptide, basically, if the side chain is acidic, like aspartate or glutamine, the average side chain pK with the alpha, the carboxyl group pKa, right? So the side chain of the acidic aspartate glutamine, if the side chain is basic, then the average is the side chain of pK with the amino pKa. For other ionizable, tyrosine and cysteine determine which is the middle pKa and average with the carboxy pKa. Okay. So in here, for example, the cysteine, the pKa, the pi for cysteine, which one is the middle one? Is A is in the middle because 10A1, A is the middle. Use the A average with the carboxy group. So it's 1.86 plus 8 divided by 2, 5.02. Okay. And lysine, so basically, the average the side chain pK with the alpha amino group pK. So lysine, that's the basic lysine. All right, so we have this carboxy 2.1, amino group is 8.95, and pK3 is 10.5. So we will average the side chain pK, which is 10.5, and the amino group 8.95. Average these two give you 9.74. Okay. And then for the peptide, so they say we have these 3 peptides, right? So you would determine the peptide from the amino group from glycine. So we have glycine amino group, all right? And then glutamine, now be the carboxy group, all right? So we have amino group, carboxy group. And then we determine the side chain, right? Add the side chain, the aspartate, that's 3.8, and also the side chain glutamine is 4.07. And then you have this, for these 3 amino acid peptide, you have this 4 number. Then take the 2 in the middle and the average. That'll be the PI. So the protein structure. So the protein structure, primary, secondary, tertiary, and quaternary structure. So the primary structure basically is just the amino acid sequence, all right? Secondary structure basically is a polypeptide backbone. They form this hydrogen ion, all right? Hydrogen ion put within the polypeptide backbone, and that give you the secondary structure, give you the alpha helix, beta pleated sheet, all right, or beta turn. So these are the primary, secondary structure. Okay. This alpha helix is right-handed, all right, and it has 3.6 residue per turn. And also you can see from this alpha helix, every number 0 amino acid and number 4 amino acid, their peptide bond, CONH, they can form a hydrogen bond. Okay, number 1, number 4, number 1, number 4, then they can form the hydrogen ion, hydrogen bond to stabilize the secondary structure. Okay, and the beta pleated sheet, okay, they also can form the hydrogen bond between these peptide bond. See the CONH with this CONH here to form this hydrogen bond to stabilize the structure. So for the parallel, so basically they both the same direction and anti-parallel, different opposite direction. And terminal, C terminal, and terminal, C terminal parallel, this is anti-parallel. Okay. And tertiary structure, basically the 3-dimensional structure of protein. They include all the secondary structure in their folding pattern. Okay. And for the tertiary structure, basically it's stabilized by the several interaction to give you this tertiary structure. So we have the hydrogen bonding, all right, between the polar amino acid. And we have hydrophobic interaction between the non-polar hydrophobic amino acid. Electrostatic attraction between the which is the ionic bond, okay, between those positive charge and negative charge amino acid. And then also you have disulfur bond between the cysteine. These are the force to stabilize the three-dimension, the tertiary structure. Okay. These are all just a combination of alpha helix and beta pleated sheet, and to give you a different type of a structure, secondary structure. Okay. And the quaternary structure basically is a multiple polypeptide chain organized in a multiple subunit protein complex. That's quaternary structure. So that involve more than one protein, okay, more than one peptide chain, and can be a homodimer, or heterodimer, right? It's quaternary structure. The fibrous protein basically is multi-subunit complex that are abundant in nature. They have long fiber or large sheet. They are mechanically strong, insoluble in water, play important structural role in nature. There's a fibrous protein, including the keratin, silk, collagen. Okay, this is silk and collagen. And globular protein, spherical protein, multi-subunit protein complex. Okay, it's a soluble, all right? So globular protein are soluble in water and salt solution. The folding, so primary sequence lead to a limited number of possible folding pathways. The free energy difference between the native and the nature must be favorable. So when we remember that in the chapter 2, the free energy must be smaller than 0, so that can occur spontaneously. So entropy change can contribute to the folding process. Okay. So the protein that facilitates the formation of stable three-dimensional structure, two types: the clamp type, which like for example Hishap protein, or chamber type. That's a GRO-EL, GRO-ES protein complex to help to fold the protein. And these are the chaperone protein, okay? So we have clamp type, which is Hishap protein, chamber type, GRO-EL, GRO-ES protein complex. Both bind to the misfolded protein, use ATP hydrolysis to facilitate correct folding. So their function help newly synthesized protein fold properly, rescue misfolded protein, disrupt protein aggregate. Okay. So the protein misfolding can lead to the disease. So for example, the cystic fibrosis, right? So the transmembrane protein, there's a mutation, right? Like misfolded protein, then you have degraded protein, those of function. The Huntington disease, so you have polyglutamine tract expansion. If misfolded protein, you have aggregated protein, so it cannot function. Okay, so basically that's the chapter 4.